Hello, I'm Heba Sailem. I'm a research fellow at the University of Oxford. I will talk about knowledge and context-driven machine learning for analyzing large-scale genetic screens. Genetic screens offers a powerful tool for characterizing gene functions. So we can use gene perturbation in order to see how it affects the cellular phenotypes. So for example, wild-type colon cells here are stained with DAPI for nuclei and green for rotavirus infection. We can start perturbing genes using siRNA or CRISPR and observe how this will change the appearance of the cells. For example, if it's increased rounding, then we can infer a role in contractility. We can knock down another gene and observe how the cells change. By doing this for different genes, then we can identify genes that have similar appearance, like very large cells here, which resemble a senescence phenotype. Once we identify genes that are similar, we can say that they are functionally linked. Of course, imaging is one way to capture phenotypes, but we can also capture phenotypes at the expression level, as well as the viability or number of cells following the perturbation. Although this tool is very powerful and widely used around the world, the current analysis pipeline of genetic screens is very challenging and doesn't provide a clear functional interpretation. For example, once we have a genetic perturbation datasets where we have multiple features describing the result of losing a gene, we can use this data and these profiles to infer what genes are phenotypically similar. This can be done using clustering, which can identify quantitatively similar phenotypes. Then we try to identify the functions associated with each cluster using functional enrichment. However, this approach is very difficult and not working in practice. So for many studies, 99% of data will not be analyzed, and only a small set of features will be focused on due to their interpretability. This means we really need new approaches to analyze these very rich data sets, especially that now we still don't know the function of a third of the human genome. To address this challenge, I developed knowledge and context-driven machine learning framework. My idea is to start from our knowledge. So start from the top. What we know about genes involved in cell cycle. We can take genes involved in cell cycle and try to find if they have distinct signature from a random set of genes. This can be done using SVM or support vector machines, which select the discriminative features and then identify separation plane. Once the classifier is trained, we can use it to predict the function of other genes based on phenotypic similarity. We can do this for many other functions. Any dataset will not be complete in terms we can capture every possible phenotype. So some classifier will not find a signature and will not be used in the prediction. We can aggregate predictions from different functions. Therefore, we have multiple predictions on possible gene functions, which can be represented based on gene ontology terms for each gene. I applied KCML to different data sets, including gene expression and imaging data. I trained it for more than 1,000 gene ontology terms. First, I found that KCML performed better on classifying gene ontology terms than random set of genes. Also, KCML outperformed existing approaches using clustering, for example, k-means or self-organizing maps. Importantly, I can validate many of the predictions by KCML based on the new annotations added to the gene ontology database, and this applies to different datasets. In summary, KCML allows us to systematically analyze large-scale genetic screens. It has many advantages as we can identify biologically relevant features, identify the phenotypes and their functional relevance, and link them to genes. Importantly, I also found that different data sets, like gene expression or morphology, can provide different functional insights, indicating the importance of integrative approaches. Thank you for listening, and I'm happy to have any questions.